And hi everyone, I'm Bingyu, and I'm now a final year PhD student at the University of Pennsylvania, advised by Dr. Yong Chen. And today I'll introducing our work on Robustify p-value calibration in observational studies with partially valid netted control outcomes. So we know real data is very important in evaluating the effectiveness and safety for medical interventions. We see this in many domains like vaccine effectiveness, SARS-CoV-2 infection, long COVID, cancer therapies, and others, but do not like randomized controlled trials. Observational studies are always vulnerable to residual bias, including unmeasured confounding, misclassified exposure outcomes, selection bias, and missing data. So even after propensity score or other adjustments, this residual bias can still often remain. So to mitigate this residual bias, netted controls have emerged as an important tool for this purpose. Netted control outcome is a clinical outcome that should not be causally affected by the treatment of interest, but share similar source of bias as the primary outcome. So if we observe an association between the treatment and the netted control outcomes, that tells us there should be some residual bias in the study pipeline. And because we can estimate this bias empirically, netted controls are not only used to do bias detection, but also for bias correction, like p-value or confidence interval calibration. And here is an example of implementation. In this Annals of Internal Medicine paper, the exposure of interest is COVID-19 vaccination, and the outcome is SARS-CoV-2 infection. Here, the netted control outcomes are events like injury, speech delay, or other events that are not related to COVID-19 vaccination. And by looking into the association between vaccination and these netted control outcomes, we can build a now empirical distribution and then use this to correct for the estimated vaccine effectiveness. But the netted control outcomes are not always valid. The current frameworks are assuming that all netted control outcomes are valid and using a single normal model, which assume that each netted control are following the same bias distribution. But this assumption can fail. Some netted control outcomes can be affected by different confounding structures, data quality issues, or coding practices. And these invalid netted controls can actually distort the null distribution. Or in other words, bad netted controls may break the calibration. So here is a simple illustration showing that why a single normal can fail. Let's see if all the netted control outcomes are from two underlying bias here in blue. And if we are using a single normal model, this red line, we can see that it cannot fit the shape very well. We have a biased estimate mean and a much larger variance here. So to address this, we proposed a more robust calibration framework here with two assumptions. First, we assume a two-cluster matrix model. One is the valid cluster, that is the true nulls, and the other captures all invalid netted controls that may have different levels of bias. And second, we assume the majority rule. More than half of the netted control outcomes are valid. And in this symmetry model framework, we observe for each netted controls have an estimated treatment effect yi with standard error si, and we assume each netted control outcome comes from one of the following two distributions. One is a valid cluster with mean mu1 and variance sigma1 squared, and the other cluster, the invalid netted control outcomes with mean mu2 and variance sigma2 squared. And this observed netted control distribution is modeled as the mixture of these two. And we can estimate the parameters using EML algorithm. So after we recover this valid null distribution, we can calculate the calibrate p-value for any new drug outcome pair. So the calibrate p-value is calculated just to plug in the treatment effect and standard error for that new drug outcome pair into the valid null distribution, which means mu1 and variance sigma1 squared. And we conducted simulation studies to compare the proposed mixture normal model to the single normal model. We vary the proportion of valid net control outcome pi from 0.7 to 0.9, the number of net control outcomes from 100 to 300, and also the separation between the two clusters between 0.6 to 1. And here, the red line shows the proposed mixture model, and the blue ones are the single nominal model. We can see here across most of the scenarios, the mixture model actually achieved the nominal type 1 error. And for parameter estimation here, the first two are from the normal model, and the remaining are from the mixture model. We see the mixture normal model produces less biased estimates. And also, when we do have true non-null effects, we found that mixture normal model are having consistently higher power when the separation to the null is increasing. We also conducted a real-world comparative effectiveness research using EHR data from Penn Medicine. We're focusing on patients with type 2 diabetes and comparing GLP-1 receptor agonists to DPP-4 inhibitors on 6 cardiovascular outcomes. We're doing large-scale propensity matching and applying a modified Poisson regression model on both primary outcomes and the net control outcomes. And this is the distribution for the net control outcomes. Here, the left panel shows the funnel plot, and the x-axis are the treatment effect, and y-axis are the standard error. And the right panel shows the density for the treatment effect. We can see actually signs of heterogeneity here. We also reported the treatment effect estimates from these 3 models, the uncalibrated model in red, the normal calibrated model in blue, and the mixture model in green. We can conclude that the mixture normal model compared to the nominal model here, it actually have a smaller bias correction and a narrower confidence interval here. And scientifically, we found that GLP-1 RAs have protected cardiovascular outcomes compared to DPP-4 inhibitors. So to conclude, real-world data enables large-scale observational research, but is vulnerable to residual bias. And net control outcomes are essential, but their validity cannot be guaranteed. So we propose a robust calibration framework that can distinguish between these two clusters and enable bias correction even with partially invalid controls, and this improves the reliability of p-value and confidence interval calibration. Yeah, and I'd like to thank all my co-authors. Thank you so much for your contributions, and open to any questions. Thank you so much.